Hi guys, welcome to A&P. We're going to be talking about development, and this is going to include sections both from chapter 28 as well as chapter 29. So let's start with chapter 29. The embryonic period extends from fertilization through the 8th week of development, and then afterwards it's called the fetal period. So fertilization means that the genetic information from the haploid sperm and haploid secondary oocyte merge. What do I mean by haploid? That means you have half the genetic material of a diploid cell. Diploid cells are most of your somatic cells, whereas your germ cells or your reproductive cells are haploid. The sperm swim from the vagina to the cervix, and then they pass through the uterus and the uterine tubes, and this is mainly because of contractions of the walls of these structures. In order for fertilization to occur, the sperm have to penetrate the corona radiata and the zona pellucida. Remember we talked about corona radiata in the previous chapter. And so basically they're going to have to get through two cell layers in order to fertilize. So the sperm, in order to do this, have a specialized set of enzymes on their acrosome. And recall what sperm look like. Basically, the acrosome is the head region of the sperm. And then the tail movement allows to push them forward. And this allows them to penetrate through that cell wall of the corona radiata. After it gets through, the nucleus of the sperm becomes the male pronucleus, and then the nucleus of the fertilized ovum becomes the female pronucleus. And recall that meiosis II only completes after fertilization occurs. So basically, as soon as that fertilization or that sperm enters, you have the female portion complete meiosis II, and then those two merge, and this process is called syngamy, and that is how the diploid zygote is formed. After fertilization, the zygote begins a series of mitotic cell divisions called cleavage. The first cell division takes about 6 hours, and then successive divisions take less time. What you see by the end of the second day after fertilization is a second cleavage that yields 4 cells. And then the cells divide again and again, so that by the end of the third day, there's 16 cells, and then each division causes the cells to get smaller and smaller. Those cells are called blastomeres. By the fourth day, the cluster of cells resembles a mulberry, and that's why it's called a morula. And it's still surrounded by the zona pellucida, and it's still the size of the initial cell, which was called a zygote. What's interesting is that fertilization occurs in the uterine tubes, and all this cell division is occurring as this zygote is moving down the uterine tubes. So the first cleavage occurs about 30 hours after fertilization. By the time it's a morula, it's 3 to 4 days after fertilization, and about 4 or 5 days after fertilization, the morula is going to enter the uterus. Okay, so then by the 32 cell stage, the fluid now inside the morula rearranges the blastomere so that it's a fluid-filled cavity called the blastocyst cavity. The mass is now called the blastocyst, so it has a new name, and it's still the same size as the zygote. So you go from zygote to morula to blastocyst. What's interesting about the blastocyst is now there's going to be two different cell populations. The inner cell mass is called the embryoblast, and that's what's actually going to develop into the embryo. The trophoblast is the outer cell mass, and it's going to develop into the outer chorionic sac that surrounds the fetus and actually a portion of the fetal placenta. So then the blastocyst is free in the uterine cavity for about 2 days, but then it implants into the endometrium of the uterus at about 6 days after fertilization. When that implantation occurs, it's usually going to be in the posterior portion of the fundus or the body of the uterus, and you can see that up on top. And the inner cell mass, which is going to become the embryo, is going to orient itself towards the endometrium. At around 8 days, we start to see some differentiation. So we get two layers of cells called the hypoblast and the epiblast. The hypoblast is eventually going to develop into endoderm, and the epiblast is going to develop into ectoderm. You start to see the amnion forming, and that is going to surround the embryo eventually and get filled with amniotic fluid. Okay, so recall we went from morula, which was just a ball of cells, to blastocyst, which was a hollow ball of cells with another ball of cells inside. The next major event that occurs at the 3rd week, and that's called gastrulation. And what happens here is that 3 different germ cell layers form. You have ectoderm, mesoderm, and endoderm. So recall back to the beginning of the semester when you're learning about where tissue types came from. You learned about these 3 different tissue types, and you were subdividing. For example, ectoderm was bone, mesoderm was muscle, endoderm was organs, and different things like that. So basically, you can see that they start to form at week 3 of fetal development. At the 4th week, embryonic folding occurs. And basically what happens is you have a flat embryo that starts to fold into a 3-dimensional cylinder. All major organs develop between the 4th and 8th week of development. And your book goes on to describe a lot of these, how the brain develops, how the heart develops, and it's pretty cool. This process is called organogenesis. You can see here a little bit more of the organogenesis process. And it continues with more and more changes going on from week 4 to 8. By week 5, you start to see rapid development of the brain and the head. And this is where you're going to see limbs starting to grow as well. At week 7, it's still called an embryo, but the regions of the limbs are becoming distinct, and digits, or fingers and toes, are starting to show. So by the end of week 8, you have eyelids that are going to be able to come together so they can close. You started out with a tail, but the tail disappears, and external genitals begin to differentiate, meaning you're going to be able to tell if this is male or female. And digits or fingers are distinct, meaning there's no longer webbing in between them. During this period, the placenta is also going to develop. Placentation is the process of forming the placenta, and the placenta is the site where there's an exchange of nutrients and waste between the mother and the fetus. And the placenta, once it's produced, is going to produce hormones that help to enable pregnancy to keep going. So how does the placenta develop? Well, embryonic tissues are going to invade the uterine walls and actually start to erode uterine vessels. So by the end of the 2nd week, we're going to see chorionic villi start to develop. Within those chorionic villi, you start to see the development of blood vessels that end up connecting to the embryonic heart. And this is going to happen by the end of the 3rd week. The vessels connecting to the heart do so by way of the umbilical arteries and umbilical vein through what's called the connecting stalk, and this is going to eventually become the umbilical cord. So the placenta is fully formed by the beginning of the 12th week, and it has 2 parts, the fetal portion, which we call the chorionic villi, and the maternal portion, which is the endometrium. The fetal period begins at the 9th week after fertilization, and what you see is that tissues and organs that develop during the embryonic period just start to differentiate, but we don't really see many new structures appear during the fetal period. So mostly every organ that you have by the end of your 8th week. Really, the fetal period is all about growing in size and adapting all those organs that were created during the embryonic period to be able to survive out of the uterus. So now let's backtrack a little bit to Chapter 28, Section 5. And I'm going to talk specifically about how the reproductive system develops. Like I said, the majority of the organs develop during the 4 to 8 week period called organogenesis. But we're focusing on the development of the reproductive system because we just finished it in chapter 28. So what you see is that the reproductive systems develop from several structures and require several chemical substances. The gonads develop from intermediate mesoderm that gives rise to gonadal ridges. And what you see is that the Wolffian ducts, also called the mesonephric ducts, develop into the male reproductive system, whereas the Mullerian ducts develop into the female reproductive system. So basically, prior to the 5th or 6th week, you have these Wolffian ducts, gonadal ridges, Mullerian ducts that are present in both males and females. So basically, you have both potential sex organs in males and females. And then they differentiate based on chemical cues, either in male or female. So in males, you have an X and a Y chromosome, and some genes on the Y chromosome stimulate the production of this hormone called Mullerian inhibiting hormone. And that prevents the development of the Mullerian ducts. So instead, the mesonephric ducts, also called the Wolffian ducts, start to develop because testosterone from Leydig cells stimulates their development into the epididymis, the vas deferens, the ejaculatory duct, and seminal vesicles. However, in females, since they don't have a Y chromosome, they do not secrete Mullerian inhibiting substance. So that means the Mullerian ducts are going to be allowed to grow. And they don't actually form testes because of this, so then they're not going to secrete testosterone. So without the testosterone, there's going to be no development of the mesonephroducts. So basically, it's just these two hormones interplaying as to whether the fetus is going to turn into a male or a female. So that's what happens with the internal reproductive organs. But what's going to happen with the external reproductive organs? Because basically, how on an ultrasound can doctors tell if you're going to have a male or female baby? Well, before differentiation at 5 weeks, all embryos have the urethral, or also called urogenital folds, urethral groove, genital tubercle, and labial scrotal swelling. And then we start to see differentiation after 5 weeks. What you see is that those structures start to differentiate. And this is pretty interesting. So at about 10 weeks, you see the clitoris, which is about the size of the glans penis. But then those start to change up until birth. These are called homologous structures. Remember, I talked about them when I talked about the reproductive system. So what you see is that the urethral folds are going to become less and less prominent in the males, whereas in the females, the urethral folds are going to turn into the labia minora and the vestibule. And then the labial scrotal swelling is going to turn into the scrotum in the males, while as in the females, it's going to turn into the labia majora. Okay, that's it. I want to give you a few assignments and reminders. So Friday is our last Zoom class, and we're going to be reviewing for the final. We've got a lot of Kahoots that we can work on together. Also, the research symposium report is due. I want to show you how to find it on Converge. Underneath the research symposium report, I actually created this extra link, which is just a survey that you have to fill out after you watch the videos. And you're going to see this little megaphone. That's the survey that you need to look for. Then one last assignment is our final. We have that Wednesday, April 29th, and like the other quiz and tests, I just open it up anytime that day. and you will have 2 hours to complete that final, and it is a cumulative final. So if you have any questions for me on that final, I would like you to either email them to me or ask them during the Zoom class, because if you have a question, another student probably has that same question. Okay, that's it for today. Happy studying, and I will see you on Friday.